This is determined by epigenetics. And this too. Do you want to learn about the therapies that modify epigenetics? Which companies are developing them? And what the future holds for them? Hop in, we are starting the taxi. We are starting today in Africa, where termites, other insects governed by epigenetics, also build their structures. If you would like me to visit your city, let me know in the comments. This is Taxi Biotech, a program to discover news and updates from the world of biotechnology and learn about the impact on the stock market. Today, I'm presenting a quick and straightforward roundup of the current state of epigenetics therapies. The stock data I show here reflects the trajectory of companies or the clinical trials and does not constitute any investment advice. This is purely for entertainment. Okay, let's start. Epigenetics is a fascinating concept that is always in vogue for biotechnology, more for its possibilities than for the therapies currently available. The fact that there are different types of ants within a single colony is the same as the various cell types in humans. This process is determined by epigenetics, which silence or express specific genes. To simplify, without diving into complex molecular details, we can say that when chromatin is highly condensed, genes cannot be transcribed. But when chromatin is accessible, the genes are available for transcription. By turning genes on or off, the function of each cell is determined. If this process is not properly regulated, it can explain the malfunctioning of certain cancers. Okay, we can distinguish between two types of drug aimed at modifying epigenetics. Generals here, or specific or precise here. General therapies. These kind of therapies modify the epigenetic machinery broadly, impacting in many genes. Multiple general inhibitors have been approved. For example, these histone deacetylase inhibitors, methyltransferase inhibitors, isocitrate dehydrogenase, DNA methyltransferase inhibitors, LSD1, and others. I have here a list that I will leave it in the comments. These modifications, methylations, demethylations, acetylations, and others, are capable of expressing or inhibiting genes involved, for example, in the proliferation of cancers. So, is epigenetics considered a success? Well, it has always held promise, but safety has been a significant drawback due to many non-specific effects. Here is a timeline showing that general medications, in orange, have been recently approved, with next-generation drug pending approval, in purple and blue. So broad gene modification can be beneficial for patients, but lacks scientific appeal. Scientists are passionate about specifically controlling epigenetics to modify genes at will and achieve targeted effects in the body. Don't get me wrong, this is a meme. I'm talking about curing syndromes like Prader-Willi or muscular dystrophy, among others, which have a high percentage of patients with well-defined epigenetic characteristics. Or simply modifying pro-cancer genes to turn off oncogenic cells into healthy ones. Okay, how it works. Direct epigenetic responses are typically achieved using a guide RNA which directs the response to a specific genome region. This RNA is paired with a deactivated Cas protein linked to repressors or activators. Okay, let's watch an animation from Epicrispr technologies. A yellow guide RNA identifies the desired region, the DNA, the protein machinery binds, and it begins to act. This protein machinery uses a rise via lipid nanoparticles. Okay, but there is another approach proposed by Sangamo Therapeutics. They use zinc finger regulators. These zinc fingers can bind to a specific DNA triplets. When several zinc fingers are linked together, they can target a specific genome region. Using repressors or activators, they can repress or activate genes. So now I'm going to talk about different companies working on different solutions. For example, Epicrispr Technologies. This company has programs aimed at restoring normality in diseases like muscular dystrophies, hypercholesterolemia, alpha-1 deficiency, retinitis pigmentosa, and blood cancers. Tune Therapeutics. This company promises fine regulations of the genes it targets. They have found that the effects on final proteins are visible even 500 days after therapy injection, indicating a long-lasting impact. Although theoretically transient and reversible, this durability may concern. If a problem arises, there is no off switch, and one must wait for the effect to fade. Regulatory agency will need long-term safety margins for approvals to ensure no undesirable off-target effects. Chroma medicine. This company's platform can open or close chromatin regions to silence or activate genes. I think that while silencing genes is technically simple, reaching highly condensed chromatin regions with guide RNAs or other molecules is challenging due to the methylation-rich environment that hinders their arrival. Here we can see that Chroma medicine is developing a drug that blocks the expression of hepatitis B virus DNA with its technology. Life Bioscience focus on specific drugs in preclinical stages for ocular and neuro repair. The science go further with technology. Moonwalk Bioscience is a company doing maps for methylated regions in single-cell analysis, a useful tool for advancing epigenetic therapies. Other companies like Volition and Inherent Therapeutics offer epigenetic marker service. And finally, let's talk about the few publicly traded companies that are involved in this sector, at least from what I have found. If there are more companies with this type of approach, please share them in the comments. Omega Therapeutics, with a system for modifying epigenetics similar to the one mentioned earlier. They recently published preclinical data showing reduced expression of the MYC gene, which contributes to tumor growth, achieving significant reductions in expression. Here in this article from Nature. They have a phase 1 trial with 24 patients where they claim their drug specifically targets MYC and achieves a reduction in its expression. Safety issues were related to the adverse effect of lipid nanoparticle injections, meaning they were not linked to the epigenetic machinery. They state that efficacy was in line with approved therapies for this disease using different approaches, such as PD-1 antibodies. They have some moleculars in their pipeline, but they are in very early stage of clinical trials. We see their trend in 2024 and is not positive. Moreover, their financials indicate that they might require funding in the short term. And the news from just a few hours ago, at the same time with the recording of this video, Sangamo Therapeutics has been approved to begin phase 1/2 trial for treating chronic neuropathic pain by inhibiting the SCN9A gene using a zinc finger via adenovirus. They report achieving a 99% reduction in gene expression, as shown by this user on Twitter. Looking at this company's pipeline, we see a broad array of trials aimed at modifying the regulation of the various genes using the zinc finger system. they also have other gene expression methods using more conventional techniques. Their partners, as you can see, are quite good. The stock chart looks promising, especially in recent months, and is gaining momentum in pre-market trading. However, like Omega, it seems they need cash. They only have enough funds to finance operations in the first quarter of 2025. This means they will need funding soon. Depending on how that funding is handled, the stock price could respond positively or negatively. Following today's excellent news, in my opinion, the chance of an imminent capital rise have increased. So, in conclusion, based on this review of the state of the art in epigenetic therapies, my personal opinion is that they are a very promising type of therapy. On paper, they are capable of achieving almost anything, and that's why I was excited to review them. Clearly, they are opening a new door for alternative therapeutics options and cures. However, it is scientifically exciting to turn genes on and off on demand to combat diseases or enhance individuals. But from a practical standpoint, I don't care whether a patient's life is changed by a highly advanced molecular machinery or by a simple chemical compound synthesized in a lab like this. To summarize, current therapies are general modifying dysregulating epigenetics in certain types of cancer to restore cancer cells to a normal state. Now, the new ones, directed therapies, are expected to be revolutionary, but are in very early stage. I was surprised by how long-lasting the effects of epigenetics are, which has a very positive aspect but also a downside, as extensive trials will be needed to determine if there are unintended off-target effects over time. With these therapies, it will be technically possible to repress gene expressions, but expressing genes will be much more challenging because it will be difficult for the molecular machinery to bind to highly condensed chromatin regions. And as always, the problem with current therapies is delivery. How to reach all cells without using viral machinery. This viral machinery can present significant toxic effects. Maybe there are some levels of hype around these therapies, but please share your comments below. Finally, among Nasdaq-listed biotechnology companies aiming to use direct epigenetic therapies, I have only found Omega and Sangamo. Both are in very early stage of clinical trials, and it is likely that they will require funding soon. So, for now, our human cells, along with those of ants and bees, will continue wondering at every moment what they want to become. This taxi ride has concluded as we move from termite mounds of Africa to the territories of Australia, a wonderful country that I love. See you in the next video. Bye!